Have you ever built a Lego set? You open the box and inside are all the hundreds of individual Lego pieces. The picture on the box shows you the final model, and with the instructions, you know how to get there. Building a model is what I've been doing as part of my PhD. Not with Lego, sadly, but what if I told you this model could help us understand diseases that steal your memories, movement, and identity? Worldwide, nearly 60 million people are living with dementia. Alzheimer's causes most of these cases. Parkinson's affects over 10 million people. These numbers keep rising, there's still no cure. To understand why these diseases are happening, we need to take a look at what's going on inside of our cells. Our cells are made from tiny building blocks, proteins and lipids. Proteins are like tiny machines made from your genes, and they do jobs around the cell. Lipids form structures like the membrane that surrounds the cell. When these biomolecular building blocks assemble correctly, the cell is healthy. And when they don't, things start to go wrong. For my research, I'm interested in a crucial system occurring in our cells known as the endosomal system. It's a sorting pathway that's disrupted in Alzheimer's and Parkinson's. It's kind of like a Royal Mail delivery service inside of your cells, where the sorting office, which would sort parcels for delivery or returns, is this floating lipid blob inside the cell, known as the endosome. The endosome makes sure essential materials or cargo get to the right place at the right time. To study this cargo sorting, I rebuild the process outside of the cell by mixing purified proteins and lipids together in a tube. It is a bit like building a Lego set with no instructions and figuring out how everything fits together. One key piece is a protein called retromer. And retromer forms these arch-like structures on the endosome to help create transport tubes that send cargo to where it's required in the cell. In Alzheimer's and Parkinson's, retromer doesn't work properly, so the cargo can't be sorted, and then the cells of the brain get stressed and die. So imagine the endosome decorated with retromer as the picture on the Lego box. This is the model I want to build for my PhD. I just need the pieces and the instruction manual. To uncover that manual, I will use a technique known as cryogenic electron tomography, an ultra-powerful 3D microscopy that will allow me to visualize retromer assembled with other proteins and lipids at the molecular level. And from this, therapeutics can be designed that specifically target and stabilize important retromer contacts that are lost in disease. So if disease is like a Lego model with missing pieces, then I'm working on the blueprint to restore the build, one tiny piece at a time. Thanks for listening.